Welcome to the course Histology, and if you just start with the word histology, histology actually is a word based on a Greek root, and histo means the tissue. If I want to have an explanation for the tissue refers to a group of similar cells that performing similar function. For example, in the heart, we have the cardiac myocytes. So cardiac myocytes are the tissue of the heart, one of the tissues of the heart. Well, when the different tissues are get together, they make an organ. So heart is an organ that made by different tissues, and one of them is the cardiac myocytes or cardiac cells. Well, when we are talking about the histology, we are actually do the study of the tissue. Logy means tissue, and the study of cells and tissue. We cannot see them directly, so we need to use the microscope. In this case, you can also think that if this is a no microscope, then this study will be anatomy. So somehow we can call the histology as also microanatomy or microanatomy. Let's go forward and see that what we will do with the tissues in the histology. The first thing, we need the classification to define them from each other. So we have classification in four categories. The first is epithelial cells. The second one is connective tissues. Third one is the muscle tissue, and finally the nervous system, the nerve tissues. And epithelial cells, they are mostly for lining and covering, and also they found these linings outside the body and inside the cavities everywhere. And the other parts that we can add to this epithelial cells are glands. That, as you know, glands to the even single cells or a group of cells or structures that they are producing and secreting protein, mucus, and lipids. So they can be endocrine, then this secretion will be called hormones, and we have exocrine, as simple as, for example, sweat glands. So when we talk about the connective tissue, we can divide it based on its fluid, like the blood and the lymph, or its supportive, which is the bone and the cartilages inside our body. And the muscle tissue is based on the appearance under the microscope, can be divided into striated and non-striated. And then striated will be skeletal and cardiac muscles, because we can see those lines. Striated means have lines. And if they are not, they are the smooth muscles, for example. We'll explain them a little bit more. And also nervous system based on the nerves, neurons, and receptors. Maybe you ask what's the difference between nerve and neuron. I will explain them also a little bit more in this introduction. Let's look at the first category. This is the epithelial cell, and we can classify them by the number of cell layers as simple, which only one cell is in the thickness, or two or more cells in the thickness, and we call them the stratified, because they are stratified in the different layers here, as you see. And also there are three basic cell shapes based on the microscopic appearance again, including the squamous, cuboidal, and also columnar. We have two examples of the columnar here, which is ciliated and not ciliated. I think you know about them from the physiology. And also, if we go to the next group about the connective tissue, as I said, they are even fluid as blood or lymph, and we have different type of cells there. It can be white blood cells majorly and red blood cells, that you also should have enough knowledge about them before starting this course, actually, to understand the histology. But the connective tissue is actually form a scaffold that epithelial tissue lies in it, and also the nerves and the muscles can be embedded in this tissue. So connective tissue is the one that holding everything. And also, if we look at the different structures from the fluid to the bone, you can see it become the hardest material also inside the body. And as I said, also muscle tissue is based on the appearance, is striated or non-striated. And as you see here, also here they put the lines here to show the striation in both the skeletal and cardiac muscles. One thing that smooth muscle and cardiac muscle have in common, if you recall, is they are non-voluntary. We cannot control them, but the skeletal muscle is under our control. So this is controlled by us. And also we talk about the nervous system. If we use the word nerve, it back to the peripheral nervous system. You know, the nervous system has two major parts, peripheral and central. And if we talk about the neurons, actually we talk about the central nervous system. And also the receptors that get the messages from the surrounding and give it to the brain. They are different type of categorizing of the nerves, and so based on the shape, based on having myelin or not, based on the function, that we try to go through them based on the histology, that the appearance under the microscope, to be able to categorize them and explain them. But before that, we cannot just take a tissue and put it on the microscope and look at it. So we need actually to go through a process of the fixation and coloring, dyeing or staining to be able to make it visible under the microscope. Most of the tissues are colorless, and then they are responding to different methods of the staining, and then we are going to see how they want to do it. First thing is the fixation. Fixation means involvement of the chemicals, and then the tissue that come from a biopsy or from after the surgery, excision surgery. Then we are prepare the tissue with this material, with these chemicals, to be able to prevent tissue for digestion by enzymes or bacteria, and then be able also at the same time to, as much as possible, keep the morphologic and also chemical characteristics in the tissue. After that, for the fixation, fixative here is the chemicals that we did talk about. Then we go to the process of heating and other chemicals to be able for the make it clear and make it visible. And after that, we are going forward for the sectioning, as you see here. Usually they put them in the paraffin to be able to use this sectioning. Sectioning means when we are using a very sharp blade, fine blade, we call it microtome. Tome means cut, micro means small, and then it can have a very thin layer that's almost transparent, and then we can just put it on the slide and send it now for the staining or coloring. And after that, we have different type of coloring for different mentality. One of the most common one we know as H and E, which is the word for hematoxylin and eosin. It's based on the acidic or alkalic structure in the tissue, and the other methods also available, like histochemistry, histoimmunochemistry. We are not going for them right now. We just want to know that as an example, that what is, for example, H and E staining is. Imagine you are looking under the microscope and you see the tissue with the different colors, as you see here, and here we have two different categories of the color. One is about the purple and the blue, the other one go toward the pink and red. Well, this is based on the acidophilic or basophilic. If it's purple and red, it's the acidophilic, it's acidic area, and it's a negative charge structure, such as we have in the DNA and RNA. As you know, this is the ribonucleic acid, for example, so the acid come in the end, we know this is acidic. And also, if it's red and pink, it's go toward the alkalic, which is also the positive charge structure, such as many proteins that we have. And this is the tendency to the dye that we called also as hematoxylin, as we have here. So eosinophil is acidic, and hematoxylin is the basic or alkalic. So we can remember that maybe we use eosinophilic or basophilic, or eosinophilia or basophilia, and eosinophilia we also can use acidophilia. Well, it was an introduction. Then we are going later toward the explanation of the different type of tissue and have some microscopic picture in this regard.